Hello students, welcome to Distinction Learning Platform, and on this platform you'll find revisions, explanation to difficult topics, quizzes, tests, a lot of stuff. So before we begin our lessons, make sure that you subscribe so that you can be updated with all upcoming videos. To make the story short, let us begin. In this video, we're talking about the Entamoeba histolytica, okay? So, but before we start talking about the Entamoeba histolytica, let's talk about the introduction to Protozoa, okay? Very, very important. So, what we're saying is this, okay? So, we need to understand to say, microbiology basically is, we know it's going to be categorized into what? Parasitoroid, bacterial, viral, and fungal. We know all that, okay? So, here we're talking about the pasteurization, okay? So pasteurization basically now is going to have two important things, okay? Unicellular microorganisms and also multicellular microorganism, okay? So what we're talking about is under what? Unicellular microorganism. So you need to understand about that, okay? So this unicellular microorganism, we are going to call them what? Protozoa, okay? So basically what we're talking about is what? Protozoa. So we're talking about protozoa, which are what? Unicellular microorganism. Okay. So this protozoa, remember, they are further what? They're going to be further categorized into what? The first one is what? Amoebas, which we'll be talking about in this session. Okay. Good example is what? Under amoeba, we know that is the Entamoeba histolytica. Okay. What else? So apart from that, we know that, okay, they are going to be further categorized into what? Flagellates. Okay, we'll talk about flagellates. Okay, flagellates like what? We know like the Trichomonas vaginalis is one of the flagellates. Okay, we'll talk about that as well. Okay, then what else apart from that? We have as well what we call ciliates. Okay, like Barantidium coli. What else? We have what we call sporozoa. So what I need what you need to understand is that, okay, we need to have the background information to say, okay, what we're talking about is this and that, so that you fully understand on this topic, okay? And we have sporozoa. Sporozoa is, if you look at the plasmodium, those are the causes of what? Malaria. So malaria is under what? Sporozoa, okay? You need to understand about that. Now, our topic will be talking about the amoeba, okay, which is the Entamoeba histolytica. But there are a lot of Entamoeba that we'll talk about, okay? So that's the overview that we need to understand, okay? That's the overview that we need to understand. Okay. Now, since we're talking about what? Amoebas, we're talking about amoebas. Okay, we're talking about these amoebas. Okay. So amoebas, basically, these are what? Unicellular simple structured what? Microorganism, which we are calling protozoa. Okay, they are very, very simple. They've got a simple structure. Okay. And if you look at these amoebas, they are further categorized into what? Okay, these amoebas, they are further categorized into what? The amoeba that can be found in what? In the intestine. Okay, we call them what? Intestinal what? Amoebas. Okay, there are also amoebas that can be found as what? Free living what? Amoebas. Okay, free living amoeba. So if you look at their amoebas, okay, amoeba, they are further categorized into what? Free living amoebas and what? Intestinal amoebas. You need to understand that. So under intestinal amoebas, now here that's where you can find what? Entamoeba histolytica. That's why you can find Entamoeba histolytica. It's what? Intestinal amoeba. That's why you can find it in the intestine. Okay. Then apart from this, there is also other amoebas that you can talk about with this. What we call Entamoeba coli. It's one of the what? Intestinal amoeba. What else? There are a lot of them. There's Entamoeba dispar, okay? There's another one, okay? There's the Entamoeba gingivalis, okay? Entamoeba gingivalis, Entamoeba Huntman, a lot of types of what? And amoebas, under intestinal amoebas, okay? There are a lot. But what you need to understand is that the most important thing that you need to understand under intestinal amoeba is what? Entamoeba histolytica, the one that we'll talk about. Why is it important to study about it? It is because it's a pathogenic what? Type of intestinal amoeba. It's the one that can cause what? Diseases and infection. But these, like Entamoeba coli, Entamoeba gingivalis, Entamoeba Huntman, all those things, they don't cause what? Infections, okay? They don't. So that's why it's important just to study about what? Entamoeba histolytica. But sometimes you need to study about what? Entamoeba coli and Entamoeba histolytica. Why studying about Entamoeba coli and maybe Entamoeba this is because the structure is similar to what? The Entamoeba histolytica. So don't let's say, let me just give an example. If you are diagnosing someone, then you find Entamoeba coli, then you mistakenly say this is Entamoeba histolytica, then that would be a problem, okay? That's why sometimes it's very, very important to know the structure of the what? Entamoeba coli, because it is so similar with what? Entamoeba histolytica. You need to understand about that. Okay, what about the free living? So free living, what does it mean? Free living, it means that those they can't be found in the body of the human. Okay, sometimes okay they can go into the human body and cause what? Infection, but most of the time they can be found what? Outside as an independent microorganism. Okay, they can survive outside like that. So what we call them? We call them free living amoebas. What are those good examples? So good examples is what? We have this one, Negrella fly. Let's just say F. Negrella fly is one of the free living what? Amoebas. Okay. What else? Apart from Negrella fly, we've got what? Entamoeba. Okay. Entamoeba basically is also one of the free living what? Organism. There's also another one which we call it what? B. medullaris, okay? Medullaris. This one, B. Barantida medullaris, basically this one is also what? One of the free living what? Amoebas, okay? But you need to understand that they are also what? Pathogenic, but they are what? They are what we call opportunistic type what? Infection. So they cause type of what? Opportunistic infection. What does that mean? So they cause opportunistic infection. What does that mean? It means that if someone is already having a chronic illness, like someone is affected with HIV, then their immune system is what? Down. If their immune system is down, then they are going to take advantage of that so that they can cause infection. So basically it means that in a normal person, basically they don't cause what? Infections. Maybe in small population, but generally they don't cause infection to what? Healthy individual without what? Chronic illness. Okay, that's why they are considered as they cause what we call opportunistic what? Infection. Opportunistic infections. Okay, let's just say opportunistic what? Infection. You need to understand about that. Okay, now since we have the background of what? Amoebas. Okay, now let's dive into the details of what? Entamoeba histolytica. Okay, now Entamoeba histolytica, we said it's a pathogenic amoeba that causes what? Amoebiasis. Now, so we need to talk about the disease that an Entamoeba histolytica causes. Although we come and talk about it under what? Pathogenesis and clinical features. But under introduction of Entamoeba histolytica, we are going to say it's an intestinal amoeba that causes what? It causes what? Amoebiasis. We'll talk about the amoebiasis in details. Okay, so it's one of the pathogenic protozoa that's going to cause what? Amoebiasis. So the first important point that we've said is that this organism causes what? Okay, amoebiasis. Okay, it causes amoebiasis. Sometimes it is called what? Dysentery. You need to understand that. So the first important point is what? It causes what? Amoebiasis. Okay, and it is what? Pathogenic. Okay, intestinal amoeba. So this is the other point that I talked about. Okay, it's a pathogenic intestinal what? Amoeba. So one that's going to cause what? It's going to cause basically infections, okay, of the large intestine. So what does it cause infection? It causes what? Amoebiasis in the what? Large intestine. You need to understand like that, okay? Intestine. Large intestine. Although it can also like go outside the large intestine and start affecting the liver. We'll talk about the extra-amoebiasis type of what? Pathogenesis. We'll come and talk about that, okay? Basically, this is an organism, or this is an amoeba that's going to cause what? Infection to the what? Large intestine. It's going to cause some ulcers that we'll talk about, okay? Most important thing, okay? Now let's talk about the epidemiology, okay? Epidemiology of what? Amoeba histolytica, Entamoeba histolytica. So what's the epidemiology? Basically, we know it was discovered in 1875, but that's not important, okay? How it was discovered is not that important, okay? So the way it was discovered it was demonstrated in the patients, a certain patient stools like that, okay? So it was discovered that parasite. So basically, it's not that important. The most important thing that we need to understand is that it's the what? Third most common cause of what? Mortality. So we're saying it's the third most common, okay? Most common. So most common cause of what? Cause of what? Mortality. So that's the first epidemiological factor that you need to understand. After what? Mortality. After what? After malaria. Okay? And what? Schistosomia. Schistosomia. So we'll come and talk about schistosomiasis. So let's just say schistosomiasis, okay? After malaria and what? Schistosomiasis. So we'll come and talk about the schistosomiasis. So this is the third most common cause of what? Mortality. After what? After malaria and the schistosomiasis. Very, very important, okay? What else? What is the other epidemiology factor apart from that? Another important factor is that it's also one of the things that has been reported worldwide, and it's going to affect about what 10% population worldwide. So what we're going to say, this Entamoeba histolytica is one of the important microorganisms, because it's affect about what 10% of population will be what affected. So 10% population affected. Okay, worldwide. Okay, let's just say the worldwide is going to be affected. Okay, and about 50% of what? 50% of cases. So 50% of cases is going to affect the 50% of population in what? Developing country, and with what? Poor sanitation. So we're saying 50% population, let's just say P in what? Developing countries, and what? What else? What did I say? And what? With what? And also countries with what? Poor sanitation. So very, very important epidemiological factor, poor sanitation. Let's just write it here. Sanitation, poor sanitation. Okay, very, very important point. Okay, so in summary, what we've understood is that, okay, Entamoeba histolytica, one of the most common cause of what? Mortality. Okay, worldwide. Okay, after what? Malaria and what? Schistosomiasis. Okay, then the second important point is what? Worldwide, it's going to affect about 10% of the population. Then the 50% is going to affect the population in what? Developing country, and as well as what? With poor what? Sanitation. Very, very important points you need to understand about that. Okay, so that's about important factors about epidemiology. You need to understand on that. Okay, because we're going to be assessed on more, especially maybe this one, the third most common cause of what? Mortality. After what? Malaria and the schistosomiasis. You need to understand that. Since we've understood the epidemiology, what else are we supposed to understand now? These parasites, the same parasite that we're talking about, it exists in what form? So morphological features, we're talking about how the parasite exists. Okay, how many forms does it only have? One form, or maybe 3 forms, 2 forms? What is that? Okay, it exists major what? Only what? 2 forms. But if you start talking about strict speaking, it's going to be existed in what? A lot of what forms that we'll talk about. But to just summarize everything, it's going to exist in what? Important what? 2 forms. Okay, which is what? The trophozoite form and what? The cystic form. But we'll talk about another form that it's going to exist as what? It's a lot of forms that we'll talk about. But just to summarize, we're saying, okay, we're saying this Entamoeba histolytica is going to exist as what? A trophozoite. Okay, in the trophozoite form, as well as what? As well as the cyst form. Okay, the cystic form. Okay, so the cyst and the trophozoite what? Form. Okay. Now, so basically, it has given us to talk about what is the trophozoite and the cyst, because you need to understand what the trophozoite is and also what the cyst what it is. You need to understand about that. Okay. So come here. So let's talk about this one, the trophozoite. So this trophozoite is one of the most important things, because the one that's going to start causing what is the one that invade the what the large intestine. Okay, so no wonder sometimes this trophozoite sometimes it is refers as what invasive what form. Okay, it is sometimes called what invasive form. Why invasive form? Because the one that causes what infection. So it's the one that's going to invade the what the large intestine. They are causing what ulcers that we'll talk about. Okay, invasive form. Okay. And sometimes trophozoites, these are the ones that are going to start what multiplying. Okay, it's a multiplicative what form as well, because trophozoites, these are the ones that are going to start what multiply and become many in number. Okay, you need to understand on that. Okay. The other important thing is that if you look at this trophozoite, you've seen how it is looking. Okay, how it is looking. So if you look at this trophozoite, the way it is looking, okay. The way it is looking, this one, if you look at this one, as you can see, this trophozoite is going to have what? A certain important feature. Okay, we'll start with what? The size. Okay, the size of this trophozoite, because size is also important. Okay, because we've got a lot of trophozoite in this protozoa organism. So you need to understand what's the range of this size of this trophozoite. Okay, so you need to understand about that. So what is the range for this trophozoite? So the range for this trophozoite, it has been estimated about what? 12 micrometer. So 12 micrometer to what? 50 micrometer. Okay, micrometer, because it changes the size like that. Okay, micrometer, but the average has to be what? 20 micrometer. Okay. So 20 micrometer, but it starts from what? 12 to what? 50, but the average has to be what? About 20. Okay, you need to understand about that. Now, if you look at again the same trophozoite, it's going to have a lot of what things. If you look at this one, you've seen this one, it's going to have this outer membrane. That's basically the cell membrane. Then it's going to have this second part here, as you can see. It's going to have this cytoplasm and also the inside cytoplasm. So the trophozoite is going to have what? How many cytoplasm? Two types of what? Cytoplasm. The one that can be found outside here is what we call what? Ectoplasm. Okay. The one that can be found here is what we call what? Ectoplasm. So now this ectoplasm now is a very, very important component of the what? Trophozoite. Why? Because the one that's going to make sure that this trophozoite move from one place to another. You can ask yourself, how does a trophozoite move from the lumen of the large intestine into the what? Into the large intestine, the walls of the large intestine. How does it do that? It moves from one place to another because of what? Ectoplasm. Okay, the cytoplasm is ectoplasm. They are going to start what? Extending to form what? Pseudopodia. Okay, those pseudopodia structures, they are going to start. Those are the ones that promote what? Locomotion of this. So in short, what we're talking about is that this ectoplasm is going to promote what? Locomotion. Apart from locomotion, what else? Also phagocytosis. Phagocytosis. Most important part. So ectoplasm is going to promote what? Locomotion and what? Phagocytosis. Phagocytosis of what? You see that the most important diagnostic feature that they see, and if you look at the trophozoite, it's going to have what? Red blood cells. How did it have red blood cells? It's because of what? These trophozoites, they have that ability of what? Phagocytosing what? The red blood cells. You need to understand that, okay? Apart from red blood cells, they also phagocytose other what? Food molecules. Okay, they are going to eat up other food molecules. Okay, because they are able to perform phagocytosis. That's about it. Ectoplasm. And if you look at the ectoplasm, it is clear like that. So it's more like a transparent, homogeneous type of what? Cytoplasm. Okay, it is clear like that. Okay. Now, if you look at the inside here, this is what we call what? What do we call this? We call it endoplasm. So we call it what? Endoplasm. So that endoplasm now, if you look at it, it has some certain things inside there. So no wonder they say it's like what? Granular-like appearance. It has granules there, okay? So it's not going to have a homogeneous type of what? Appearance. It contains granules. So it's granular type of what? Appearance, okay? Now, what is the major function of the endoplasm? So the endoplasm now is going to contain a lot of structures, organelles. It's going to have mitochondria, it's going to have the nucleus, it's going to have the endoplasm reticulum, it's going to have all those things, okay? So it's a very, very important part of the what? The trophozoite, okay? So now, since we're talking about the endoplasm, let's talk about what? This structure, if you look at this one, this is what we call a nucleus. Now, so you need to understand something about the nucleus, okay? And under a nucleus, we talk about a lot of things under a nucleus, okay? So we'll talk about the three important terminology that they use. Don't get confused, okay? So three important terminology that they use under a nucleus. What are those? Okay, three important terminology. So they use what we call chromatoids. Here I need to explain about this three important what terminology. So since we know this, I can remove this. We know that's endoplasm and everything there. So, okay, this is chromatoid. So that we are going to use chromatoid. They use another one. Okay, they use another terminology which is chromosome. Okay, another one that they will be using in future lessons. We are going to be using what? Kinetoplast. Okay, we're going to be using this. Okay, so you need to be using, if you talk about the nucleus, you need to know the words chromatoid, chromosome, and what? Kinetoplast. If you are understanding that word, basically it will be very, very easy for you to understand. Okay, so now ask yourself, what is a chromatoid? Okay, what is a chromatoid? A chromatoid basically is what? Basically it is what? It is the DNA that's going to be what? Present outside the what? The nucleus. Okay, sometimes they say the extra nuclear chromatin. If they say extra nuclear chromatin, basically it's a DNA that can just be found not inside the what? The nucleus, but outside the nucleus, but within the cytoplasm. The cytoplasm. Okay, so here we are just saying extra. So the chromatoid, basically are just talking about the DNA which is outside the what? The nucleus. So no wonder if you've seen something like chromatoid bodies like that, they're just talking about those bodies contain the what? The DNA, but not within the what? The nucleus, which is very simple. Okay, very simple. So here we're saying extra nuclear. Okay, extra nuclear what? DNA. Okay, not within the what? The DNA like that. Okay, then what about the chromosome? We know the chromosome now, it's the type of DNA that's going to be found inside the what? Nucleus. So we are saying DNA within what? Nucleus. So DNA within what? Nucleus. That's what we call what? Chromosome. Then kinetoplast now, this one. This kinetoplast is non-DNA what? Or they are saying this, they say not non-DNA, we say non-nuclear DNA, okay? And this can be found just outside the what? The nucleus. So non-nuclear what? DNA found what? Outside the what? Outside the what? The nucleus. Okay, you need to take something, okay? So I need to clarify something. So if you look at the kinetoplast and what? The chromatoid, but they both found what? Outside the nucleus. But chromatoid is a DNA that can be found within the nucleus, but now it is found outside the nucleus. That is the chromatoid. But kinetoplast is a DNA that can be found outside the nucleus, but it's not the same as what? The nucleus. The type of DNA that is which is outside the nucleus is not the same as what? The one that can be found inside the nucleus. The nucleus is just something else, but it's also something that is having information or genetic information. No, we are just saying non-nuclear DNA. Non-nuclear DNA, we are saying is not the DNA that can be found within the nucleus. Okay, you need to understand like that. So these are the three important terminology that we'll be using. But here, if you talk about the Entamoeba histolytica, it's going to have what? It's going to have the chromatoid body, and it's going to have what? The chromosome. So it's going to have these two important what? Things. If you talk about the chromosome, the way it is situated inside the what? The trophozoid. the chromosome, that DNA within the nucleus, how is it situated? It is situated what is centrally. So what am I saying? Within the trophozoite, this chromosome, okay, within the trophozoite is what is centrally what is located. Okay, centrally what is located. That's how the chromosome is going to be what like located, is centrally. Okay, because there are other chromosomes that can be situated what peripherally within the nucleus, but peripherally. But this one, under trophozoite, under Entamoeba Histolytica, the chromosome is what centrally located. One of the important morphological feature. Okay, so we've talked about the trophozoite. Then apart from this that we've talked about, there is also another thing that you need to understand. This one, okay, this is the red blood cell. So, which is one of the important feature that you need to understand. These trophozoites are able to engulf, or they are able to phagocytose red blood cell. And what did I say about this red blood cell? If you find them in the trophozoite, diagnostic feature of the Entamoeba Histolytica. Okay, what do you think are these? Okay, what do you think these? So they are going to contain what? Also vacuoles. So those are what we call vacuoles. Okay. Sometimes they say glycogen vacuoles. That's why you can find glycogen within those vacuoles. Okay. Very, very important. Okay. So now since we are done with that, let's go to another morphological feature. Okay. We'll go to the cyst morphological feature here. Okay. But let's talk about this one. So what do we call this one? We call this one as what? The precyst. Just from the word pre, before the cyst, okay? So if these trophozoites want to change into the cyst, it has to pass through the precyst. You need to understand like that, okay? So this precyst now, remember that the precyst, okay, for you to understand like that, okay? We're talking about the Entamoeba Histolytica. If you remember, the life cycle is more like all the forms they change. So they change from one form to another, from one form to another. The trophozoite can be converted into what? The cyst, okay? And as well as what? The precyst can be converted into what? The cyst. So it goes like this: trophozoite, precyst, then cyst, like that. Okay, that's the way it is. Then again, cyst can be converted back into what? The trophozoa. We talk about the life cycle. Don't worry about that. Okay. So just know that now, if this trophozoite want to change its form into the cyst, it will pass through this transit stage, okay, then so that it can be fully matured into what? The cyst, okay. Now if you look at this precyst, as you can see, it's going to have different structures, as you can see, okay. Remember here I didn't talk about what? The chromatoid body. I just talked about what? The chromosome. On the precyst, that's why you are going to see what? These two important structures. What do we call this? These are what we call chromatoid bodies. These are the ones that are going to have what? DNA, which is outside, okay? Now, there is also another important thing that they have. They are going to have what? These glycogen vacuoles here that we talked about, they are going to become large like that. They are going to become large. Why? Because they are preparing for future. If that precyst now converts itself into what? The cyst can go in the environment outside the body and survive for longer. But you are going to ask yourself, how does it survive? It uses what? Glycogen, so that it can have what? Use that as source of what? Energy and what? Food. So no wonder this is going to have what? Large glycogen vacuoles. Then apart from that, now it will be converted into what? The cyst, okay? After some times now, it will be converted into what? The cyst. Now this cyst now is going to have, as you can see, it's going to have this cell membrane there, okay? The important feature of the cyst is that it's going to have what? A trilaminar type of what? Cell membrane, because going outside so that it can be protected by that trilaminar structure. Okay. So what are we saying? If you look at this cell membrane, as you can see, there are three. 1, 2, 3. Okay, there are three. Then that is what? A trilaminar. Okay, trilaminar what unit? This is a trilaminar what? Trilaminar cell membrane. This says SM or CM. Trilaminar cell membrane. So for the precyst, as you can see, didn't have what? This trilaminar cell membrane. For it to be fully converted into what? The cyst, it has to develop what? The trilaminar cell membrane. Okay. What else? Now here, as you can see, if you look at here now, this one is going to have now the just a single what cytoplasm. Okay, it's going to have a single cytoplasm like that. Okay, so this is basically the cytoplasm. Okay, and basically, as you can see, these what we call this, they are what? These are what we call what? Nucleus. Okay, these are what we call nucleus, but they are about what? 4. You need to understand about that. Okay, they're about what? 4 nucleus in the what? In the cyst, or the mature cyst. No wonder sometimes it is referred to as what? Quad. Okay, it is referred to this word quadrinucleate. Sorry, quadrinucleate. Okay, quadrinucleate what? Cyst. Okay. Quadrinucleate cyst. Why? Because it's going to have, quad means what? 4. It's going to have what? How many types of what? How many nucleus? It's going to have what? 4 nuclei. Okay, it's going to have 4 nuclei. So that is quadrinucleate what? Cyst. It's going to have that. But if you look at this one, the pre-cyst is going to have 1 nuclei. Then after some time, it's going to start dividing up to it reaches what? 4. If it reaches 4 now, it has become what? The cyst, like that. Protozoa is going to have what? 1 nucleus. So here, under the protozoa is going to have what? How many nucleus? 1. Okay? Yeah, so those are the... Now, if you look at the cyst, the other important thing about the cyst is what? Size. Okay, remember here we talked about the size. Then we said it is about 12 to 50, but basically the average is about what? But what about this one? The cyst is smaller than what? The protozoa. Basically, it's going to have 80 somewhere to 12 micrometer, like that. Okay, that should be the size of the what? The cyst. Okay. Okay, so now we are done with the morphology. Now let's go to the important part. Okay, so let's go to the important part. What is the important part now? The important part is what? Life cycle. Let's talk about the important part now. What is the important part? Life cycle. So that's why we are going to fully understand everything that I've talked about. Okay. So the life cycle of what? Entamoeba histolytica. So now, the first important thing is that you need to understand the what? The infective form of the what? This Entamoeba histolytica, because we've talked about the 3 important what? Or let me just say 2 important what? Morphological features. We've talked about the trophozoid. We've talked about the what? The cyst. Which one is an infective form? Basically, if we say an infective form, what do we mean? It means that a form that can what? That can be easily transmitted, okay, and cause infection. Because if you get the trophozoid, let's just give you an example. If I maybe get the trophozoid like that, I eat the trophozoid. If I eat the trophozoid can transmit the infection. Why? Because it will be killed in the stomach. The stomach is not infective, okay. But if you get that, if you eat the cyst. If you eat the cyst, basically now it's going to what? It's going to cause now infection. So what am I saying? The first important thing about the life cycle, you need to understand about what? The infective what? Form. The infective what? Form of the what? Of the parasite. The infective form of the parasite we are saying is what? The cyst. Now let's just specify, because the cyst, we've got a lot of cysts, so we're going to say mature what? Cyst. Mature cyst. So mature cysts, those are the effective what? Form of the what? The parasite. So those are mature cysts. And mature cysts, they've got what? How many nucleus? 4 nucleus. Okay. You need to understand about that. Okay. Now let's talk about another important thing. So now the question is, how does someone going to get that infection? Okay, that's how does that someone going to get this cyst from outside the environment? Because we know that the cyst is going to be found outside the environment and the environment. So how do they get this cyst? So they get them through what? What we say, maybe you are eating something that is infected with the cyst. Okay, it's contaminated with a cyst. You are eating that. Okay, basically the mode of transmission of this type of the parasite is what? Oral route. Okay, fecal oral route. Okay, so this one, someone is going to what? Ingest something with what? Contaminated with what? Mature cyst. That's how someone is going to what? Get the infection. Okay, so the mode of what? Mode of what? Transmission. Let's just say mode of transmission is what? Let's just say fecal oral route. Okay, that's how the what? The transmission is going to be what? Transmitted. Okay, like that. the infection is going to be transmitted via what? Fecal or what? Food. Someone is going to get that infection through what? Eating like that, okay? If they eat that cyst, so this is the mature cyst, if they eat that, okay, then it reaches into the what? The stomach. The moment it reaches the stomach, what do you think is going to happen? Here there is what? Hydrochloric acid. Hydrochloric acid will try to destroy this what? This mature cyst, but it will fail to destroy that mature cyst. Why? Because it has what? The trilaminar unit. So it's going to have this trilaminar cell membrane. So because it has this trilaminar cell membrane, basically it's not going to what? To die. It will continue its what? Transmission. It will go to continue it's not going to die like that. Then reaches the what? The small intestine like that. But basically, if it reaches now the distal ileum, basically we know that the small intestine may have what? Duodenum, jejunum, and what? Ileum. But the moment that cyst reaches what? If it reaches the distal what? Ileum here. If it reaches the distal ileum, what do you think is going to happen? Now in the ileum there is what? Alkaline what? pH, and there is the presence of what? Trypsin. So in the ileum here, okay, in the ileum here there is what we call trypsin. Trypsin will start breaking down this what? Cyst. Okay, trypsin is going to start breaking down this trilaminar unit like that. Okay, so trypsin will start breaking down this trilaminar unit so that it can become something else. So it will be just like that. Okay, it will be just like that. Okay, it will remove that trilaminar unit so that it becomes something. What is that something that we are talking about? So it will be converted into something. What is that? So we call it what? Metacyst. Okay, metacyst. So the moment this mature cyst reaches the what? Reaches the distal ileum. Okay, if it reaches there to find trypsin and there is what? Alkaline pH there. Then the trypsin will start breaking down that trilaminar unit or part of the mature cyst. Then if it breaks down like that, it will convert that mature cyst into what? Metacyst. Okay, so now so if you look at here this diagram, we are saying because of the trypsin and alkaline components, they are going to convert this cyst into what? Metacyst. So let us write here, we are going to convert them into what? Metacyst. Okay, now do you know what will happen since we are here? So this is something that is coming like that, the metacyst that is coming like that. Okay, it will go to what? Where? What is the first part here? That's what we call what? The cecum. It will go here in the cecum there. So this metacyst, it will go into the what? The cecum. Now you know what will happen. Then the metacyst, remember that the mature cyst, how many nuclei did it have? It was 4, right? Also, if you look at here, the metacyst is going to have what? 4 nuclei. Why having 4? Because there's no nuclear division there. Now, for this metacyst to be converted into something that we need, the nucleus is going to start what? Dividing. Okay? So I'm saying the moment it reaches the cecum here, the moment the metacyst reaches the cecum, okay, or the colon there, it will be converted into something. Okay? It will be converted into what we call it? It will be converted into what we call metatrophozoid. Okay, metatrophozoid. And metatrophozoid is going to have how many nucleus? So if it is 4, then to undergo the nucleus will start undergoing what? Division. Then up to what? It's going to form about what? 8. Okay. So for the metacyst to be called a metatrophozoid, nuclear division has to happen. So the nucleus, one nucleus will undergo what? The division like that. So that this single cell is going to have about what? How many nucleus? It's going to have about what? 8. Okay. Then this 8 nucleus that we're talking about. Okay. So let's just imagine to say in the colon here, in the colon or the cecum, in the cecum we've got about what? A single cell, a metacyst like that, a metatrophozoid with what? How many nucleus? They're about what? 8 like that. Okay. Then what will happen is that each nucleus is going to contain its own what? Cytoplasm. Okay. So before I go any further here, so let us talk about what will happen to this metacyst. So I'm saying this metacyst is going to be converted into what? Metatrophozoid. Okay, metatrophozoite. Okay, how many nucleus did this one had? 4. What about this one? 4, and this one is going to have what? 8. Okay. Then what did I say? After having 8 nuclei, what do you think is going to happen? Now it will be what? Each nucleus is going to be surrounded by its own what? Cytoplasm. Okay. So each nucleus is going to be converted into what? It's going to have its own what? Cytoplasm. So let's just get 4. Okay. This one, that one, that one, that one. Okay. Another one. 1, 2, 3, 4, like that. That one, that one, that one, that one. Okay, like that. So that's what will happen in the what? In the large intestine, in the colon there. Okay. So what are we saying? So we are saying to say, okay, this one is going to be converted into what now? A trophozoa, mature what? Trophozoa. So let's just say mature trophozoa. Sorry, having what? Just one nucleus, and they are going to be about what? One nucleus, and they are going to be how many mature trophozoa we are going to have? They are going to be what? About 8 what? Mature what? Trophozoa. Mature trophozoa. Each with what? A single what? Nucleus. So each with what? One nucleus. So let's say 1 end. Okay. So mature trophozoa, they are going to be about what? 8. They need each trophozoa is going to have what? A single nucleus, because we said here a trophozoa, a mature trophozoa has to have what? One nucleus. You need to understand about that. Okay, so now this happens now. Okay, now we have this mature trophozoid. We have this mature trophozoid here. Okay, then what they will start doing is like this. Okay, this mature trophozoid, what do we say? These are what? Invasive type of what? Trophozoid. They will start now invading the what? The mucosa of the what? Of the large intestine. They will start causing ulcers here. Okay, they will start causing ulcers here. This is mature trophozoid, and these mature trophozoid, these are the ones that will start what? Dividing. They're active, so they will start what? Dividing like that. So this one will divide into another one. They will divide another one. Okay, they will divide into another one. So that they can become large in number. But it has been shown that this division that I'm talking about, they are going to divide every after what? 8 hours. So this trophozoid will divide so that they become 2. Again, they divide, they become 4 like that. But every after how many hours? 8 hours. Okay, and they divide by what? Binary what? Fission. We'll talk about that as well. Okay. Now, if it finishes whatever it's going to be doing, okay, it will be transported like that. Okay, it will be transported like that. So this mature trophozoids will be transported, but it will sense now the moment I'm being transported like that. If I'm not invading like that, I'll be left in the room like that. So what I'm going to do is that then it will be transported like that, transported like that, up until outside. But it will sense that okay, a trophozoid can't survive outside the body, because if the moment it is removed from the what? The large intestine, it can't survive for longer outside the body. Okay, then what's going to happen to that mature trophozoid? Now it will start changing. It will change into something. Okay, what did I say? It will change into what? This trophozoid now it wants to survive outside, so it will change into what? The precyst. So it will say to say, okay, me outside I can't survive. Then this one is going to change into what? The precyst. Okay, it's going to change into the precyst. Then the precyst, before going outside, it will change into what? Mature cyst. Then the mature cyst is the one that will be what? Outside, and what? Survive. You need to understand about that. Okay. Again, the cycle will be what? Will be repeated like that. So this mature cyst now will be what? Will be like ingested like that. Then the same process will happen. Now you need to understand something here. So we are saying, let's complete this one. We are saying, okay, now this one is going to be converted into what? The precyst. Okay, now the precyst is be converted into what? The cyst. Yeah. So now understand something. I need to talk about the two important processes that you need to understand. You've seen this process where the cyst is going to change into what? The metatrophozoid. This process where the cyst is going to be changed into the metacyst and into the metatrophozoite. What do we call this process? We call it what? So this side we call it what? Excitation. Okay, we call it what? Excitation. Okay, so that excitation, that's what we call is where the cyst is going to be converted into what? Metatrophozoite. You need to understand about that. Okay, then basically this process where the what? The mature trophozoite is going to be converted into the pre-cyst and eventually into the cyst. What do we call that process? We call it what? Encystation. Okay, what do we call this? So we call it encystation. Okay, encystation. Okay, you need to understand these two important what terminology. So we are saying, just to recap, excitation is what? The cyst is being converted into what? The metatrophozoite. Encystation is where the mature trophozoite is converted into what? The cyst. Okay, these are the two important what processes that you need to understand in case they ask you. Okay. Okay. So now that is all about what? The life cycle of the what? The Entamoeba Histolytica. Okay, so in the next video we'll talk about the pathogenesis and clinical features, as well as what diagnosis and what treatment. That will be in the what next video. Stay tuned, start more. Make sure that you like the video and don't forget what to subscribe.